Hello everyone, this is me Zahur Islam. So today I will tell you about the bioequivalence study. So what is bioequivalence study? Before studying bioequivalence study, I will define the equivalence. Equivalence is a relative term that compare the drug product with respect to a specific characteristics, function or defined set of standard. In equivalence, we will compare the drug products with respect to the specific characteristics or the function with any standard ones. Another one is chemical equivalence. Chemical equivalence means when two or more drugs products they contain the same active ingredient and the same amount. There are many examples. For example, Voltrols. Voltrol also contain diclofenac sodium 50 mg and urine also contain diclofenac sodium 50 mg. It means here the two drugs products contain the same active ingredient in the same amount. In pharmaceutical equivalence, two or more drug products are identical in strength, quality, purity, content uniformity and disintegration and dissolution characteristics, but may differ in the terms of excipient used. In pharmaceutical equivalence, whenever there are two or more drug products, they are same or identical in their strength, in their quality, and their purity and its content uniformity and disintegration and dissolution characteristics may differ in the term of excipients. You can only differentiate it on the basis of excipients. Therapeutic equivalence, when two or more products that contain the same therapeutically active ingredient eliciting identical pharmacological effects and can control the disease to the same extent. Another important term is bioequivalence. So, bioequivalence can be defined as the drugs in two or more identical dosage form reaches the systemic circulation at the same relative rate and extents. So, for example, their plasma concentration time profile will be identical without significant statistical difference. How we can claim that two drugs they are bioequivalent? So, in bioequivalence, the drugs in two or more identical dosage forms, they can reach to your general or systemic circulation at the same relative rate and extent, is we can say that their plasma concentration time profile will be identical without significant statistical difference. What are the needs and objectives for bioequivalent studies? It is clear that if a new product is intended to be substitute for an approved medicinal products as pharmaceutical equivalent or alternative, the equivalence with this product should be shown or justified in order to ensure the clinical performance. Types of bioequivalent studies. Types of bioequivalent studies, the first one is in-vivo studies. So, in vivo study, the oral immediate release product with systemic action. So, in vivo bioequivalent study, there should be a narrow therapeutic margin, there should be a pharmacokinetic complicated by absorption, non-linear kinetics, pre-systemic eliminations, while the unfavorable physicochemical properties and documented evidence for bioavailability problems, no relevance in vivo data available. There is non-oral immediate release products and there is modified release product with systemic action. Studies are conducted by pharmacokinetic and dynamic method, which I have already explained in my previous lecture. In vitro type of bioequivalence study is biowaivers. So, in vitro studies we will check the dissolution studies. So, the dissolution studies can be used as alternative to an in vivo bioequivalence under certain circumstances. So, new drug products differs only in strength of their active substances it contains, provided all the following condition holds. Pharmacokinetics, they are linear and the qualitative composition is same. The ratio between the active substance and excipients is same, or the ratio between the excipient is the same. Both the products, they are produced by the same manufacturer at the same production site. The drug product has been slightly reformulated or manufacturing method slightly modified without affecting bioavailability. The drug product meets all the following requirements. So, it is in solution or solubilized form. The active ingredient is in the same concentration as the approved drug products. The products contain no excipient than the effect absorption of the drug. An acceptable in vitro dissolution and in vivo with similar in vitro dissolution route rate is approved medicinal product. The product is intended for topical administration, oral administration, but not intended to be absorbed. In experimental study design, the basic design for the study is determined by the scientific question to be answered, the nature of the reference material and the dosage form to be tested, the availability of the analytical methods, benefit, risk and ethical consideration with regard to testing in humans. Types of designs are completely randomized design, randomized block design, repeated Myers and crossover and carryover design, and latent square design. Completely randomized designs, all the treatments are randomly allocated among all the subjects. Method of randomization, so label all the subjects with some number of digits like 1 to 20, such as randomly select non-repeating numbers from these labels, subject them for the first treatment and then repeat for all other treatment. So, I mean you can also conduct different design studies, protocol, and sometime we are doing randomized control trial. So, in randomized design you will have to perform activity like this which I have mentioned. There are certain advantages of completely randomized design. So, it is easy to construct, can done with the number of treatment and subject and simple to analyze with variable sample size for each treatment. Disadvantages is applicable limited treatment with homogeneous subjects. Now, what is randomized block design? So, randomized block design is the subjects having similar background characteristics are formed as block. Treatments are randomized within each block which are independent of each others. There are certain advantages. The first merit is we should systemically. The systemic way of grouping provides substantially more precise results unlike completely randomized design. So, treatment need not have equal sample size. The statistical analysis is relatively simple and design is easy to construct. Repeat Myers crossover designs. It is the kind of randomized block design where the same subject serve as a block, same subject utilized repeatedly, so called a repeated Meyer design. The administration of two or more treatment one after the other is specified or random order to the same group of the patient is called crossover designs. What are the advantages and disadvantages of repeated Meyer crossover? So, the first advantage is it has good precisions and it has good economical statuses, can be performed with few subjects, useful in observing. In repeated Meyer crossover, it can the effects of treatment over the time and the same subjects. Now, you can see here the disadvantages are the order effect due to position and treatment order, carry over effect due to preceding treatment and washout period necessary 10 elimination half life. So, you can see here this is subject and this is period 4 1 2 3 4 and here the period is 1 2 A B B A. Types: replicated designs 2 formulations 4 periods 2 sequence, non-replicated design 2 formulation 2 periods and 2 sequence crossover design. Another one is latent square design. In latent square designs, all other above designs are continuous trial. However, latent square design, each subject receive each treatment during the experiment. It is 2-factor design. 2-factor design means the rows is equal to subjects and columns is equal to treatment. So, you can see, so you will divide it 2-factor design. The rows and rows you will the rows will be used for the subjects and columns will be used for the treatment. Advantages: there are certain advantages of latent square design. So, advantages are to minimize the variability of the plasma profile and carryover effects. So, small scale experiments can be carried out for pilot studies and possible to focus on the formulation variables. Now, some demerits which are also found here: there is less degree of freedom, randomization is complex and long time study, more formulation, more complex study and subject dropout rates are high. You can see these are groups 1, 2, 3, 4 and these are the subjects are grouped. So, in which different digits they are given in different different order 3, 8, 9, 4, 18 and this is treatment for page number. This is page 1, this is page 2, this is 3 and this is 4. And page 1 we have taken A, B, C, D and then page and then treatment for page number 2, this is D, C, D, A and then D, A, B, C and then page 4 C, D, A and B. This is clear. This is latent square design model. You can see here these are groups, these are subjects and these are the treatment for the page number. So, this lecture was related to the bioequivalence study. Inshallah, in my next lecture, we will talk about the methods for the enhancement of bioavailability. Thank you so much for watching my lecture. So, if you didn't subscribe my channel, so kindly subscribe my channel and press the bell icon for my upcoming lecture. Thank you so much.